My third and final figure for this exam was studying the different interpulse intervals of the male and female Photinus greenie fireflies, which I will refer to likely as P. greenie fireflies to kind of shorten that up a little bit throughout the course of this explanation. This was one of the primary figures in this particular experiment, and the purpose of it was to analyze the interpulse intervals of the P. greenie fireflies in response to different ambient air temperatures. This experiment took place in Lincoln, Massachusetts during June and July of 2004. The data was recorded in 15-minute recorded 15 minutes post sunset for about 45 minutes or until the males stopped flashing. During this time, the firefly flashes were recorded and a polynomial regression line was calculated in order to match the data to the recorded just interpulse intervals at different temperatures of the fireflies. This polynomial regression line was controlled for temperature at 21.1 degrees Celsius and is shown in purple in the figure on the screen. This figure ended up showing that a longer interpulse interval is associated with lower temperatures, likely because the chemical reaction that has to take place to allow for the flashing to occur takes place at a slower rate when the temperature is lower and a higher rate when the temperature is higher. And so therefore, the faster the reaction, the lower or the smaller the interpulse interval. We likely think that the ambient air temperature is a cause because if it wasn't, the polynomial regression line would not adequately represent the data as it does here. The second figure in this paper was studying the average male interpulse interval during 3 courtship stages: the warm-up stage, the patrolling stage, and the dialoguing stage. This was used to determine if the IPI variation was due to these courtship stages or due to temperature, kind of a comparative study. In order to get this data, they observed 3 clear stages during the male IPI mating rituals. The first one was the warm-up, which occurred directly after flight on top of a blade of grass. The second was patrolling, which occurred while the male was flashing during flight, trying to identify a female. And finally, the third was dialoguing, when a male was flashing with a female, directly having a dialoguing conversation, basically. After this whole definition of the patrolling stages, multiple males were observed for each stage, and 3 side-by-side box plots were created. What they found is that patrolling has the most variable data. However, it did have a smaller sample size at n equals 4, which could be a large factor in terms of its larger variation. Warm-up and dialoguing had less variable information, but all in all the plots centered around 1.5 male interpulse intervals measured in seconds, which means there's no significant difference between the 3 different courtship stages. In order to fully understand this data, it just means that different stages likely did not lead to the variable IPIs. Because the average interpulse interval is pretty consistent across the 3 different stages, we can't attribute the variation in interpulse intervals we've seen throughout this experiment to the courtship stage. Instead, it's more likely that it's attributed to the temperature.